There are over 1 million scientists in the US, each one dedicated to solving life's greatest mysteries. Close to 300 of them spend most of their day in this building, the Sidney Frank Hall for Life Sciences at Brown University. Today, we will explore the day in the life of a researcher at the Renan Laboratory, which uses Drosophila melanogaster, the fruit fly, as their model organism. What we have right here is an unprecedented look at these Drosophila scientists in their native habitat. It appears that most of their day, they sit in front of computers, sifting through published literature, and when they're not destroying their eyesight from staring at the monitor for too long, they look at minuscule objects through a microscope, or they play around with what appears to be various liquids at their lab benches. Or they dance.
I have colonies.
What is this sorcery? Working in the lab isn't supposed to be fun.
Well, it's fun when you get results.
Do go on.
I used fruit flies to investigate this new Talen technology.
Talen stands for transcription activator-like effector nuclease. They are proteins that were identified in bacteria that cause plant diseases. Talens consist of a nuclear localization sequence that tags the protein for import into a cell nucleus, amino acid repeats that help it bind to DNA, and a nuclease, an enzyme that upon dimerization produces a double-stranded break in the DNA. Each amino acid repeat corresponds to one base pair in the DNA. We can design custom talens that can bind virtually any DNA sequence. When a pair of talens come together and the nuclease can combine, targeted cutting of DNA occurs.
I want to integrate the Talen into the fly genome, which has never been done before. I want to use it to remove a large part of the fly genome, more specifically, the gene for the red eye collar. All the mutant flies in our lab were made using a technique that leaves behind a short synthetic byproduct that flanks the gene for the red eye collar on either side. This byproduct is known as the LOXP site, and this sequence has a very interesting property. It's palindromic, meaning it's the same forwards as it is backwards, just like the word race car. This unique property allowed for a very easy Talen design. I would only need to design one Talen that would bind the LOXP in 2 different places.
In our fly system, we have the gene for red eye collar flanked by 2 LOXP sites. I want to design talens which will bind in those sites outside the red eye gene. Designing just one Talen, the palindromic sequence, allows me to have 2 Talen pairs that will bind to DNA and enable the nuclease to come together, or dimerize, and therefore cleave the DNA, essentially popping out the eye collar gene. Losing this gene causes the flies to have white eyes instead of red. You can see just how easy it is to spot this color difference yourself.
Talens have never been expressed from the genome before. So far, researchers have only achieved temporary Talen expression by injecting Talen mRNA into the genome of their model organisms. Having a constantly active Talen can therefore be a very valuable tool in genetics. With this research project, I hope to report whether talens can be used to excise a large genomic sequence, as well as what their specificity and precision in cutting is. My findings will help us better characterize this technology, and hopefully propagate its use in Drosophila genetics. Without a doubt, talens can become a new and valuable tool for genome manipulation.